Okay, so I'm going to talk about biosimilars in oncology and where we're going with that, because it is a relatively new area. These are my conflicts of interest. So biologics, as you heard, represent a large portion of the pharmaceutical market, and also, as you heard as well, a huge amount of money that's spent internationally, although that money is disproportionately spent in the developed world. Clinical care, we'll hear about supportive care with myeloid growth factors. There's also, of course, erythropoietic stimulating agents. And then active therapy with monoclonal antibodies, antibody drug conjugates, and small molecule inhibitors or stimulators and cytokines. So there's a lot of different therapies that we're using in oncology now, and targeted therapy has, of course, made a huge inroad into the treatment of a variety of different solid tumors. And now we're using antibodies in the area of immunotherapy as well. So it's been really a huge advance in oncology. But what about these agents? Well, as you heard, these reference biologics can have variations over time, and I'll show you an example of one evaluation of this, which is quite difficult. But manufacturing changes can cause this shift over time, process changes, and also just location changes. So if you make a biologic in one country versus another, you move your manufacturing place, that may change the agent a little bit as well. We know that biosimilars are not identical to reference biologics, but what we want them to do is to have similar within our acceptable range clinical activity, immunogenicity, and safety. Generics are actually identical. As you heard earlier, you can just take a chemical structure and reproduce it. What you stick along with it, our patients will often say, well, that pink pill made me have this symptom, but the white pill didn't. There's obviously different binders and in specific agents. Somebody gave an interesting poster on that yesterday. So biosimilars are not generics, and Maddie showed you this earlier. The different pathway is listed here for the U.S. guidelines, and as this is actually an international guideline, but the numbers that are at the top are the international FDA numbers, 351K pathway. And what I did was highlight clinical studies, because that's what I'm going to talk about. Because in terms of clinical use of biosimilars, really where oncologists end up and pharmacologists is looking at the clinical data. There's an enormous packet that's submitted to the regulatory agencies of hundreds and hundreds of pages that represent those first three areas of the triangle: analytic, non-clinical, clinical pharmacology with PK/PD. And it includes that PK/PD in clinical pharmacology studies in healthy volunteers, where you're comparing the biosimilar to the originator product. And in fact, in the case of trastuzumab, it required studies that compared both the U.S.-made trastuzumab to the EU-made trastuzumab, because it was thought there might be variations there as well. So that was also very valuable information. As Maddie mentioned, there may be drifts over time, and as I mentioned, based on both processing changes, different manufacturing sites, different countries. And this is actually really fascinating data that was done really to look at a biosimilar trial with a biosimilar trastuzumab, and there was a suggestion that actually the biosimilar could have been a bio-better, so actually resulted in better pathologic complete response rates in a neoadjuvant clinical trial in HER2-positive breast cancer. So they went back actually and looked at the different lots. And what you can see actually, if you look at first drift and second drift, is they looked at a variety of different factors. So one is the analytics. So you're looking at the amount of sugars, so mannose, and galactosylation, which is basically putting sugars onto the antibody. And what you can see is that there actually was a shift in terms of the relative area over time in a first and second drift. And then there's also, we don't really have good in vitro tests to evaluate the activity of a biologic agent. So that's one of the problems. Trastuzumab's always been a big issue. You don't have a good preclinical animal model and preclinical studies. But you can look at antibody-dependent cellular cytotoxicity and Fc gamma receptor 3A binding. And what you can see is the activity really did shift over time. Do we know that that's clinically important? No, but we do know that there was a shift over time. And that's actually really important. And because of this data and because of biosimilars, there's actually tighter regulations now, an overview of the biologics overall to try and make sure that there isn't so much shift over time. But processing really does make a difference. So what do we have for clinical requirements in order to have biosimilars enter the clinical space? So you want to have the pharmacokinetic study for establishing bioequivalence. As I mentioned, that's usually in healthy volunteers, but you could also have an early small study in patients as well. And then you have one clinical study, so at least one study. And one of the issues about the requirements of those studies has been that it's a bit unclear. So there's been a variety of different ways that these studies are done, as I'll show you in just a moment. The regulations and the guidelines suggest that you should do this in the most highly sensitive population. So a population where the drug really works well is the idea. And you want to use a short-term endpoint to try and establish similarity in efficacy, and at the same time collect immunogenicity and safety data. And then if there's residual uncertainty, you could do additional clinical trials. But intriguingly, in the field of oncology, using trastuzumab as an example, additional clinical trials have not been necessary because the trials were so overwhelmingly similar in their results, and they all looked very much the same. So there are a number of different oncology biosimilars that are approved in the United States, with the most recent trastuzumab oncology biosimilar approved this month. You can see actually that there are 5 different trastuzumabs starting in December of 2017. There is a bevacizumab, and then there are a lot of different supportive care medications and one rituximab. So we're actually behind Europe on the use of biosimilars. The patent laws are completely different. So one patent in the EU might be much longer than in the US, and another one might be much shorter. So how patents are done is beyond my level of exploration because it's so incredibly complicated. And it's resulted in a number of different legal battles about the use of biosimilars and what actually constitutes a patent, which is a fascinating area beyond the scope of our discussion, but many of these have settled in the United States, which have paved the road for the use of biosimilars in the US, where they're already in active use in Europe, outside of the supportive care area that you'll hear about next. You know, it's interesting, supportive care biosimilars, the first one we had was filgrastim, and, it took a little while, but maybe a few months to a year, and then the institutions just sort of took these on and said, we're only going to use the biosimilar, and we're not going to use the originator product because you're going to sell it to us at a cheaper price. So this became, it became like using the word Kleenex for tissue, right? Is that we use the word, the brand name for the biosimilar as filgrastim now. I'm always trying to bring people back to filgrastim, but that's what it's become. And nobody really questioned if you have a little less efficacy or a little more, it didn't really matter. But when you talk about using biosimilar cancer therapeutics, particularly in the curative setting, there's a much higher bar for acceptance on the part of both oncologists and patients. Although I have to say, with our biosimilar growth factors, the patients never blinked an eye. I mean, they really said, if you think it's okay, we're okay with it. So we'll talk about the clinical trials in just a moment. So the goal of these comparative clinical studies is to exclude any clinical relevant differences between the biosimilar and the reference product, understanding that these agents are not identical and that we don't require these for agents that are identical in chemical structure. The biologics are made in living cells, and so there are differences. There are always going to be differences in how you produce them, and then the post-translation, the sort of translation of the agent into the product you give, varies considerably. Extrapolation, you heard about a little bit earlier, and immunogenicity studies are all very important concepts in the use of biosimilars. And it's actually fascinating to think about immunogenicity, in that I had never really thought about it, and we just sort of accepted that, trastuzumab, bevacizumab didn't cause antibodies, There were no publications that really went through that in any detail. Actually, most of the data now comes from the biosimilar studies in terms of whether you make anti-drug antibodies. So the patient criteria, of course, is to live longer. You want to live longer with your cancer, and so that's the primary goal. Disease criteria for clinical trials that are more pragmatic are response rate, disease-free survival, progression-free survival, or pathologic complete response. And then for biosimilar clinical trials, if you want to look at a short-term endpoint, you look at response rate. That's our quickest, shortest-term endpoint. What you want to know is that endpoint is going to correlate with longer-term endpoints. So if you're going to use response rate, which is what's been chosen for all of these trials, then you just want to know that that endpoint in general correlates with either disease-free or progression-free survival. And of course, you'd like it to correlate with overall survival as well in previous studies using the originator product. And then you need to, of course, allow understanding of safety and immunogenicity. So you have to give the drug for long enough to know that you're getting reasonable results. So what's the optimal clinical trial setting? It very much depends on the agent that you're using. So for example, if you were going to test bevacizumab in lung cancer, you would choose a different endpoint and a different trial design. You're still going to use response, but you might not need quite as much because you're not looking at survival endpoints in that group of patients. But for trastuzumab in breast cancer, it's become a very, very big question because trastuzumab is given in early and late stage HER2-positive breast cancer, and it helps people live longer. More people are cured of HER2-positive disease when they get trastuzumab, both in early stage disease, and then we have cured patients with metastatic breast cancer with HER2-positive disease. Not a huge number, but some. So it really makes a big difference. So how should you test this? Well, the beauty of neoadjuvant trials is there's a short-term endpoint of pathologic complete response, which is response rate. And you can give a 1 year of drug exposure, so you get safety and immunogenicity. Disease-free and overall survival are such long-term endpoints, it really adds to the expense of the trial, so it's much more difficult to do. And post-surgery treatment may impact your endpoints, particularly now that we have agents that we can give that change outcome in the post-surgical setting. First-line metastatic trials, when they're done in patients who don't have access to these agents, patients are generally treatment naive. They get long drug exposure for the majority of patients who are responders and continue to respond. You have a highly sensitive endpoint of overall response rate. You can evaluate progression-free and overall survival over a relatively shorter period of time. And overall response rate has correlated with PFS and OS in HER2-positive disease, just like pathologic complete response has correlated with long-term endpoints in the early stage setting. So we actually have two very good paths that are great and have reached regulatory approval for looking at biosimilarity of trastuzumab. We know that trastuzumab is a very active drug that improves outcomes, as I mentioned earlier, in both metastatic and early stage breast cancer. And also it improves outcomes in metastatic gastric cancer that overexpresses HER2. And it's the gold standard of treatment in HER2-positive cancers. In addition, trastuzumab is pretty well tolerated. We understand the toxicity well and can generally manage it, and we understand how to screen. So the first actually trial to be published that led to regulatory approval looking at a trastuzumab biosimilar was the Heritage trial, which actually was a many year development and putting into action trial, mainly because this was sort of an unexplored new area. And of course, the patent wasn't going to run out until many years in the future when we first started this work. The trial was designed in the first-line metastatic setting, and when the trial was first designed, they were giving it to people in first and second line. But of course, that wasn't really a great way to look at the endpoint. So after the first 50 patients, we redesigned the trial as a first-line study. Patients received taxane of choice with either the originator trastuzumab or the proposed biosimilar at the time. And then after patients had a response in clinical practice, we dropped the chemo and we continued the antibody alone. So that's what we did after 24 weeks in our evaluation of the primary endpoint of overall response in this trial. So we reported the best overall response rate at week 24, our primary endpoint in the intent to treat population. As you can see, that was about 450 patients. And the EU and US have slightly different endpoints, either the ratio or the difference in overall response rate. Both of those fell well within the required range. So basically, you set up a range that you think is reasonable. When this data first came out, there were a lot of questions about whether or not the ranges were reasonable for clinical efficacy. And so we've been able to actually show that these are very good ranges developed by our statistical colleagues. So you can see the pre-specified equivalence margin on the far right, and that this number fell well within that margin. So then we also looked at secondary endpoints, progression-free survival through 48 weeks. So this is actually a really interesting endpoint because what you're looking at is you had a patient who was responding or had stable disease at 24 weeks. You drop the chemo and you just continue the antibody alone. And what we showed was identical progression-free survival. You can see that the confidence intervals are also almost identical in this patient population. And at that time, at 48 weeks, our overall survival was still immature. And that actually is a very, I think, poignant point about biosimilar work. What that means is that women with metastatic disease that's HER2 positive benefit tremendously from access to these antibodies. And all of these people on this trial would not have had good access to the antibodies without the trial itself. So the trial these trials have affected a huge number of people worldwide. So initially here, this just shows you the curves of the progression-free survival, and our overall survival looked very good but was immature. And then we also wanted to show that you could correlate the short-term endpoint with the longer-term efficacy endpoint. So we looked at progression-free survival at week 48 and the correlation with overall response at week 24. And what you can see is very tight correlation between these endpoints, as you can see in both the responders and the non-responders. So it really validates the use of these short-term endpoints. We showed that safety was very, very similar. More patients receiving the trastuzumab DKST, as it's known now, had some myalgias. Actually, this shows you hematologic toxicity because they received paclitaxel, which is kind of interesting. And the cardiac toxicity was identical, as well as adverse event-related deaths, and they weren't due to antibodies. During monotherapy, patients do very, very well. They have minimal toxicity, as you can see in the far right-hand side, and there was no difference between the DKST or the originator. Immunogenicity is fascinating. There are some anti-drug antibodies, but they're very low, under 3%. And it seems like people had antibodies when you started, at baseline, which was kind of intriguing. And it didn't really increase over time, and it had no impact on outcomes. So this is actually very helpful information. At ASCO this year, just a few weeks ago, we presented our overall survival, our final analysis of overall survival at 36 months. So this is now 3 years in a patient population before trastuzumab who had a overall survival median of less than 2 years. And what you can see is overall survival is identical, and that many women are indeed still surviving far out who had access to trastuzumab. At 36 months, we actually, the company actually transitioned everybody who was on originator trastuzumab to the biosimilar product for cost reasons, so that patients who are still responding continue to have access to the antibody until they no longer need it for whatever reason it is. We have no idea how long to treat patients who don't progress, but generally we in the U.S. treat them for 10 years. So I put the data together compared to the historical data, showing that our overall response rates are very similar. The time to progression is very similar to what the previous studies, the pivotal trials that led to approval. And immunogenicity, as reported, is also relatively identical in terms of antibody-drug antibody rates over time. So it really proves our whole idea of biosimilarity and led to the first approval of a biosimilar trastuzumab in the U.S. in 2017. Although biosimilars are not yet in clinical practice in the U.S., we expect that to occur, we hope, later this year. So there are a number of studies. The rest of the majority of the remaining trastuzumab studies have looked at pathologic complete response as a primary endpoint. This is CTP6 compared with trastuzumab in the neoadjuvant setting, and then continuing the proposed biosimilar for 1 year with chemotherapy in the induction before surgery. What was shown was the total PCR rate was identical with CTP6 compared to the reference trastuzumab. The primary endpoint was total PCR, which is what we use as a correlate to long-term outcome in HER2-positive breast cancer. And they looked at the difference as well as the risk ratio, and it was also identical, and safety and immunogenicity were identical as well. In addition, there's been updates subsequently that have shown that safety in monotherapy is identical in addition, and that the 1-year disease-free survivals are also similar. This shows you the 1-year data that was presented at San Antonio last December, and of another trastuzumab biosimilar called SB3, also used the neoadjuvant setting to test this and showed identical PCR rates, but they now have 30 months of treatment, also showing no difference in event-free survival in this patient group in the early stage setting. So very, very good. And what you can see is the event-free survival is almost 97%. So these patients do very well when exposed to the biosimilar. One of the questions that's come up is whether or not you can combine this biosimilar trastuzumab with the antibody pertuzumab, which is approved in the U.S. and Europe as neoadjuvant and adjuvant therapy for HER2-positive breast cancer. And because there's no interactions between these antibodies, we feel that there's no issue with combining with pertuzumab. The countries that participated in these neoadjuvant and metastatic trials didn't in large part have access to even trastuzumab, so they certainly weren't going to give pertuzumab to these patients. But I think that the combinations are fine, as well as combinations with other chemotherapy drugs. I mentioned there are now 5 approved trastuzumabs listed here in the United States and also approved in Europe. Pharmacovigilance is an interesting area. There's a mandate for pharmacovigilance, which is following the safety of these drugs afterwards, but there's no very good path. Like, nobody said this is what you should do. You're supposed to report any unusual toxicities, but hopefully over time, we're relying on our pharmacology colleagues to help us with pharmacovigilance. And really, the idea is that this will help us also understand whether we can use it as interchangeability, so you could change somebody from one trastuzumab to another without being concerned about toxicity. We talked some about extrapolation earlier. Interchangeability is a big area, and then naming. And I thought I'd just show you the naming here, that the FDA designated suffix must have no recognizable meaning and 4 letters and be lowercase. But you will notice that filgrastim sandoz looks very much not like that. And that is because filgrastim sandoz, SNDZ, was named before the FDA designation for biosimilar suffixes. So they're the only one that gets that name. It's kind of funny. So the goals of the biosimilar clinical trial program are to demonstrate similar efficacy and safety compared to the reference product, and not to reestablish benefit, but actually these trials have reestablished benefit, and they've shown biosimilarity, and now we have a number of different options to potentially lower the cost to the healthcare system, but also improve access to people, which I think the biosimilars are already doing worldwide. I was in Algiers talking about biosimilars, where already the cost savings over 2 years has been staggering, allowing access and money to be used in other important areas. And also, I think competition is important, and having a variety of therapeutic options is important as well. Thank you.